Okay, welcome everyone. Thank you for joining us. This is our third ICSA/yICSA webinar, and for those of you who didn't have the chance to attend the previous meetings, this series has been set up as a way to take the place of those events that had been organized during the year and have had to be postponed due to the current situation. So for the next few months, we will have a series of talks every couple of weeks from both early career researchers in the field as well as more senior researchers. And we really welcome new people joining. So if you would like to contribute and share your research with us, please do get in touch. We are hoping to keep them running at least until next year when we meet in person at the ICSA conference in Osaka next year, hopefully. So today we have two very exciting talks. We have Emanuel from the University of Edinburgh, and we have Eleanor Jane Tyler from the Barts Cancer Institute in London. If you have any questions during the talks, I will please ask you to wait until the end of the talk and then just raise your hand so we can unmute you and you can ask your questions directly to Philip or Ellie. So with that, I will stop sharing my screen and I would like to introduce our first speaker. Give me a second. Hi, Philip. So Philip is a final year PhD student at the University of Edinburgh. Before his PhD, he completed his undergrad in his hometown of Newcastle upon Tyne, where he then worked for 2 years developing biosensor assays for diagnostics. So his current research involves, involves using models of acute lung injury to investigate alveolar epithelial senescence and how these cells may have a wider impact upon the tissue repair processes within the lung via the SASP. So the aim of his research is to highlight senescence as a therapeutic target in human acute respiratory distress syndrome. So today he will be talking about modulating type 2 alveolar epithelial senescence in acute lung injury and fibrosis. So thank you, Philip. Off you go.
Okay. Yeah, hopefully everybody can hear me okay. Thank you very much for the introduction, Star. And thank you very much to all of the yICSA committee for giving me the opportunity to come and talk to you today about my PhD research, which is on modulating type 2 alveolar epithelial senescence in acute lung injury and fibrosis. So I thought I would just start off by introducing a bit more about myself and my background. So as Stella mentioned, I'm a teacher at the University of Edinburgh. So it's been my great pleasure to live and work in the capital of Scotland with our very own castle and extinct volcano. And to explain how my PhD project came about, it's perhaps appropriate to introduce my PhD supervisors. So my primary supervisor is Kev Dhaliwal, who is a respiratory consultant and a chair of molecular imaging and healthcare technology. And his real focus is on pushing translational healthcare technologies into clinic. And my second supervisor is a slightly grainy David Fehrenbach. I apologize, David, that I couldn't find a higher res image to steal off the internet. But he's a senior nephrologist who has a research focus in cellular senescence, in particular aging diseases of the aging kidney. So it's really through a collaboration between these two that my project came about, which is looking at senescence in the lung. In particular in the alveolar epithelial cell, and whether this could be a viable therapeutic target in the acute respiratory distress syndrome. So to introduce my project, rather than talking about all the features of senescent cells, as I'm sure most of you are well aware of, I thought I would introduce the alveolar epithelial cell, which is a remarkable cell that resides in the epithelial cell layer, in the deepest region of the lung, in these sort of bunch of grape-like structures. The alveoli, which are little air sacs that are surrounded by capillaries to facilitate gas exchange into our blood. So the epithelial cell layer is 1 cell thick, and it's made up of 2 different key functional cell types. So the type 1 alveolar epithelial cell, which is a long, thin, flat cell which covers a vast surface area and facilitates gas exchange. But interestingly, there's also these cuboidal cells, these type 2 alveolar epithelial cells, which are perhaps best known for producing pulmonary surfactants, which stop the alveoli from collapsing in on themselves. But interestingly, they have a number of antimicrobial properties and also act as progenitor cells within the lung. So following injury, they actually proliferate and de-differentiate and redifferentiate in order to repopulate this crucial cell layer. And although this was quite a nice image to demonstrate, so it's an electron micrograph where this false colored cell in the middle here is a single type 1 cell sat on this bed of capillaries. And it really shows how fragile yet robust these cells have to be, as they're often the first to bear the brunt against infection and so on. And it's accompanied by these type 2 cells as well. So next, I'll quickly introduce the acute respiratory distress syndrome. So ARDS in this case isn't age-related diseases, as it is in most senescence talks. But in my title, I've mentioned acute lung injury, which isn't really a term that's used in clinic. It's more of an umbrella term. Whereas patients will be diagnosed with acute respiratory distress syndrome, which is still, there's a lot misunderstood about it, but it can be characterized into various phases. So here's a schematic of a healthy alveolus with the type 1 and type 2 cells separated by the interstitium from the surrounding capillaries. And in the early exudative phase of ARDS, we see the influx of neutrophils into the airspace and the breakdown of this crucial epithelial cell layer, which causes the influx of pulmonary edema in the lung. As this cell begins to repair, we often see widespread fibroproliferation in the lungs, which inhibits their ability to return back to 100% functionality. So hopefully, in most patients, it will resolve, but it's actually through this prolonged or chronic fibroproliferative phase that these patients are reliant upon mechanical ventilation. And this is actually the phase in which you see the highest mortality in patients, which prior to the COVID pandemic was at around 40%. So there's clearly a need for more biological therapies in ARDS and more understanding, as it's still heavily reliant on preventative measures such as prone positioning of patients or protective ventilation. So I'll not go into this diagram in a huge amount of detail because I'm sure you've seen many like it before, but this is really, for my project hypothesis, is built around the idea that if these type 2 alveolar epithelial cells that are required for repopulation of the epithelium are becoming senescent and entering into proliferative arrest, and furthermore, if they have a secretory phenotype that can exacerbate inflammation and fibrosis, then this could potentially promote aberrant repair and fibrogenesis following acute lung injury. And for my project aims, can really be summarized in 3 short questions, essentially. So can we suppress it? So this was looking at neutrophil epithelial interactions and what the effect would be on TGF-beta mediated fibrosis and ultimately senescence. Can we drug it? So looking at this ever-emerging class of compounds, senolytics, if these cells are causing issues, can they be pharmacologically depleted? And finally, can we image it? So again, with a push on translational technologies, whether we can develop fluorescent compounds for imaging and monitoring senescent cells in tissue. So for the talk, we concentrated on my second aim, looking at some of the work that I've been doing with Senolytics. So I'll start off by introducing some in vitro work using a model of type 2 alveolar epithelial senescence, where I use A549 cells, an AT2-like cell line, injured with bleomycin to induce senescence. So anybody who has worked with these cells before or knows about alveolar epithelial cells, I'll introduce the elephant in the, I'll address the elephant in the room that these cells are an adenocarcinoma cell line, and so they do have specific mutations in the KRAS genes and P16 gene, for example, which will affect their proliferation in culture and essentially makes them senescence resistant. But there's still viable data to be gained from this model because they do have features of AT2 cells, such as the production of pulmonary surfactants, etc. So it's a very basic model where basically I treat with bleomycin for 2 hours and then culture under normal conditions for 5 days and then continue with analyses. And one of the first analyses that I carried out was XGAL staining for the presence of senescence-associated beta-galactosidase, which is characterized by this typical blue cytoplasmic staining. We can see these cells also growing in this sort of large flattened shape compared to the confluent monolayer that we'd expect in the non-senescent cells. And furthermore, when I went to test different dosages of bleomycin, you can see that hopefully you can see that there's an increase in the number of blue staining cells, and also the cell density was more sparse across the bottom of the wells. And when I came to quantify these, the proportion of cells occurred in a highly repeatable and dose-dependent manner as I went through increasing concentrations. So as I mentioned before, bleomycin acts by directly damaging DNA and causing DNA double-strand scission. So it was assumed that this would be inducing senescence through a DNA damage response. So to confirm this, I carried out immunocytochemical detection of the DNA damage marker phospho-H2AX. You can see in the nuclei of these senescent cells this characteristic fluorescent foci of phospho-H2AX when compared to the non-senescent cells. And as I also mentioned before, the cell density is far more sparse in the senescent cells, suggesting they were entering into proliferative arrest. And when testing for P21 in the nuclei of these cells, you can see a vast increase in the proportion of cells that were P21 positive. And when using an image processing algorithm to test for how many cells were strongly fluorescent of P21, you see it increased from below a quarter to above 3/4 of cells. So the next thing I wanted to look at was what the SAS profile of these cells was. So I looked at a selection of mRNA transcripts and quantified them within these cells. And it showed that there was an increased expression of P21, which agreed with some of my previous staining data. And conversely, there was a decrease in the proliferative marker Ki67. I also looked for a selection of pro-inflammatory cytokines and saw that there was vast upregulation in IL-1 beta, IL-6, and CXCL8, which are commonly cited as being key components of the pro-inflammatory SAS. Finally, there's various increases in procollagen 1, procollagen 3, and TGF-beta pro-fibrotic factors, which suggests that these cells could be having some sort of interplay with surrounding immune cells or fibroblasts. So the next thing I wanted to look at is whether these cells can be selectively depleted using Senolytics. So again, slightly tailored the model, where after the 5 days in normal culture, they were incubated for a further 3 days in the presence of drug, and then the viability was analyzed. And I initially looked at D&Q, so 50 micromolar quercetin with various concentrations of dasatinib, and the results were fairly underwhelming. It was highly variable, and it was only at the higher concentrations you could perhaps start to tease out some sort of preferential toxicity. However, when I looked at navitoclax, ABT263, looking at a low, medium, and high concentration, these were far more promising results. So it was far more consistent and showed that it was selectively toxic towards the senescent cells rather than these non-senescent cells. So next I did an Exon5 PI testing for the presence of early apoptotic cells, because BCL2 a family of protein inhibitor, and so the idea is that it drives these cells into mitochondrial mediated apoptosis. So I looked at an earlier time point, so this was just looking at 6 hours following incubation with drug, and then analyzed by flow cytometry. And I thought I would just show a couple of these forward scatter and side scatter flow cytometry plots, just to show how heterogeneous the senescent cell populations were compared to the non-senescent cells. And it took a little bit of persuading for the flow cytometry text that these were indeed the same cell type, just somewhat injured and somewhat. So when looking at PI and an Exon5 FITC fluorescence, you have the senescent cells along the top and the non-senescent along the bottom there, looking at 0, 0.55, and 10 micromolar of ABT. You can see that in the senescent cells, the population begins to smudge over into this third quadrant here. which suggests that they are entering into apoptosis. And the non-senescent cells aren't completely unaffected, so we can see that there's this population starts to emerge at the higher concentrations. So there is happening here, but I apologize that these percentages are so small, but it's around 50 to 60% at the higher concentrations in senescent cells, and it's below 5% in the non-senescent. So the conclusions that I can take from this was that the model was working. So it was effectively and consistently inducing senescence in these cells in a dose-dependent manner. But interestingly, they were entering into P21-mediated proliferative arrest, and as I mentioned, they adopted a pro-inflammatory and pro-fibrotic SASP, which suggests that they could be contributing towards this overwhelming inflammatory reaction that's occurring within the lungs in vivo. And they are nicely depleted, selectively depleted by ABT263 in vitro, and all of this warrants further testing in vivo. So next I'm going to describe some of the initial characterization work that I've been doing for an in vivo model of alveolar epithelial senescence. So again, using bleomycin is a well characterized historical model of acute lung injury when instilled intratracheally. And I would go so far as to argue that it's perhaps a more appropriate model of ARDS than IPF as it's commonly used, because following bleomycin injury, after a number of weeks, actually start to see resolution of fibrosis in the lungs, whereas IPF is characterized as being irreversible. So C57 black mice were received either 24 micrograms of bleomycin intratracheally or vehicle control. And I'll try and explain the injury time course progression here with some picrocereus red staining of these lobes of lung, where collagen is stained red. So at day 7, we see a small amount of difference. At higher magnification, you do see an influx of inflammatory cells. But at day 14, hopefully you can see with these images, we start to see a difference as there is fibrotic areas starting to crop up in this patchy manner. And it appears to peak at some point between the day 14 and day 21 time point. And then by 28, there is still residual fibrosis, but again, it appears to be a bit more evenly spread across the lobe of the lung. So the next thing that I wanted to look at was detection of markers of senescence in alveolar epithelial cells. But to do that, I needed to work up some assays that I can effectively detect the cells that I was interested in. So as a marker for type 1 cells, I used a transmembrane glycoprotein called aplanin, which is present on the membrane surface of epithelial cells throughout the body, but one of which is type 1 cells. And as a type 2 marker, as I mentioned, these cells are surfactant producing, so this is prosurfactant protein C. And I was happy with how this worked out, because you can see there's membranous staining within the alveolar structures, but not within the epithelial cells of the bronchioles and larger airways. And furthermore, if we look, you can see that there's breaks in the membranous staining between these type 1 and type 2 cells. So that's just another example there that showed that it was working quite nicely. So looking at the DNA damage marker phospho-H2AX again, this is just an example where I looked at type 1 cells and phospho-H2AX. Because bleomycin in vivo has a similar, has the same mechanism of action, where it induces DNA damage. And you can see that there's a slight amount of background staining in the vehicle controls. However, in the bleomycin injured animals, you can see these fluorescent foci start to come out in the nuclei of these cells. So the next thing that I wanted to look for was P21 in these cells, as P21 is more commonly being cited as mediating pulmonary fibrosis and being a key player in senescence within the lung. So this is looking at an early time point. So this is 7 days post-injury prior to fibrosis. And I must explain that I've switched up the colors a little bit here. So these green stained cells are the type 2 cells. So this is for the pro-SPC cells, And then when I looked for P21 nuclei, I saw that there was widespread P21 expression and a vast difference between the vehicle controls and bleomycin. And interestingly, it appeared that it was almost exclusively expressed within these double positive type 2 alveolar epithelial cells. When looking at a later time point, when fibrosis has started to establish, it gets a little bit more complicated and started to see a mixture of double positive cells, and more commonly these P21 positive cells that were not type 2 cells, and then as well the type 2 cells that were not P21 positive. And when it came to quantify these, the proportion of type 2 cells that were also P21 positive appeared to gradually decline when looking through the later time points. So this suggests that there might be some level of immunosurveillance, or these cells are being cleared as the time course of injury goes on. But I think one of the most interesting things to take from this is that there is that widespread expression prior to fibrosis occurring. So some of the conclusions I could take from this initial characterization work, I know that the next logical step is then to administer ABT263 in this injury model and see what the effects are. Unfortunately, literally the week that I was about to start those experiments, the UK lockdown rules came into enforcement, and so all non-essential research had to be stopped essentially. So I'm disappointed that I can't present that data to you today because I haven't done the experiments yet. But what I can take is that intratracheal bleomycin induces P21 expression increasingly in AT2 cells, but prior to pulmonary fibrosis. And would this be reduced by ABT263 administration? And what would the knock-on effects possibly be for the amount of fibrosis that we see in the lungs? Whether these senescent cells could be acting as a protective mechanism, or whether it could be detrimental. So to investigate that, I plan to in future carry on with an in vivo experiment that I've designed, where I'm looking at the early time points, so administering ABT as an intervention just a day or two after injury, and also looking at a later time point as well, where we'd expect peak fibrosis to see what the effect of this might possibly be. Okay, so see what I'm doing for time now. So just as a final slide, I'd quite like to just present some of the work that I've actually been starting since lockdown has came into enforcement. So in light of the COVID pandemic and ARDS induced by COVID-19, therapies are more needed than ever. And people are well aware of that, especially with the recent news over the past couple of days with the dexamethasone treatment on patients that are on mechanical ventilation. So I'm sure that you've seen lots of graphs like this, where the mortality in COVID-related deaths is much higher in the aged population, which suggests there could be a link between cellular senescence and people who do succumb to COVID-19 infection. So in Edinburgh, we have an excellent tissue resource in place in the BioQuarter, where my lab is based. So there's a huge interdisciplinary effort that's been ongoing for the past few months. And there's a number of trials up and going as we speak. And one of which that I'm involved with is the ICSA, we're basically working with one of the pathologists from the hospital who's been carrying out postmortems on retrieving samples from patients that have died from COVID-19 induced ARDS. And the idea of this is really to try and develop some of these immunofluorescent assays that I presented today, and try and ask the question of whether there are increased levels of alveolar epithelial senescence, particularly in type 2 cells present in these patients that succumb to COVID-19 infection, and comparing them to healthy age-matched controls to see whether there'd be any difference in that. So I'll finish there. I'd like to just say thanks to the funders and to once again to ICSA and Young ICSA for inviting me and allowing me to present today. acknowledge everybody in the Alveolar group and the Fehrenbach group. I was looking for a recent sort of picture of the lab group socializing, and I came across this one. I thought was quite appropriate because I think everybody can relate to having Zoom drinks or whatever over the past few months. So yeah, that's me. Thanks.
Thank you, Philip. That was very nice. If anyone has a question, I think Marta has one. Let me see if this works. Marta?
Yes, can you hear me?
Yes.
Okay, perfect. Hi, Philip. That was a great talk. Thanks so much. I really liked kind of how you pointed out that people usually use a bleomycin model for IPF, but maybe it's actually a more appropriate model for ARDS, like you were saying. And I was wondering, I assume the majority of the work that you're doing is in young, healthy mice, right?
It is, yes. So it's all 10 to 14 week old male mice is what I've been working with.
I wonder if you've looked or if you had plans to look in older mice, because what I know about the model is that actually the fibrosis induced by bleomycin in old mice is permanent. So it's irreversible, much like it is in the age population that gets it. So I would be really curious to know whether the AT2 cells are implicated there or whether it's an acute kind of phase because of the installation in the young mice specifically.
Yeah, that's a really interesting question. I mean, the answer is no, that I haven't looked in older mice. It's something that would be great to do in future. But I guess in the interest of not trying to answer too many questions at the same time, it would be interesting that you say that it's irreversible in older mice, because again, that suggests that if these senescent cells that are induced by bleomycin are persisting, and are not being cleared by the immune system as they might be occurring in my model, then yeah, that would be a really interesting question to try and answer.
Yeah, and sorry, one other thing that I wanted to ask, and you might have mentioned this, so sorry if I missed it, but so you see the senescent AT2 cells decrease in the later time points, like from day 21 to 28 or whatever, and you start to see some non-AT2 senescent-like cells emerging. Have you characterized what cell type that is? Are those fibroblasts, or do you know?
No, so that's something that I plan to do with the samples that I have. So yeah, it would be really good to look for a fibroblast marker, because I think that it does occur within these areas of fibrotic tissue as well. That I start to see the emergence of these cells. So I think it would be really interesting, because then it might suggest that there's some sort of paracrine crosstalk between the AT2 cells and yeah, as you mentioned, fibroblasts, which I suspect. You know, I think it's important not to keep the blinkers on too much and just focus on one cell type and try and take in the whole sort of you know, inflammatory environment that will be going on between these cell types.
Okay, great, thank you.
Okay, well, I have a question as well. It's a bit related to what Marta just said. You mentioned that there was a slow decline in the burden of senescent cells, right? Like from first week towards the
just looking at P21, I should mention, but yeah.
Okay, I wanted to ask you, because you also suggested that there might be an immune clearance, right? Have you looked at the immune compartment? Like what's the profile of immune cells that you have recruited?
Yeah, so I could actually, I do have actually no, I'll not bother showing the extra slide, but yeah, basically what I do is at each time point I've been taking bronchoalveolar lavage samples as well, just to look at the what cells are present in the air spaces. And at day 7, you see a vast increase in the amount of neutrophils, which suggests that, again, it's largely neutrophil mediated, some of these injuries that are occurring. But again, that seems to rapidly drop off at the later time points as well. And again, this is the only data that I have, but it's the vast majority is alveolar macrophages. It's mostly alveolar macrophages that I will sort of retrieve through these methods. Again, I think more immunofluorescent staining for perhaps looking at NK cells or any other cells that might be involved in senescent cell clearance would perhaps be the better way to approach that. But that's not something that I've done.
Okay,